Dr. D. Sangeeta is currently a senior lecturer at the Department of Chemistry, Anna University, Chennai. She has completed her PhD in the field of polymer chemistry from the University of Madras in the year 2000 and has teaching and research experience of more than 10 years. Her research interests are in the following fields: fuel cells for clean energy, functional membranes for desalination, and polymers for biomedical applications. She has published more than 20 research articles in national and international journals and has more than 40 research articles in national and international proceedings. Presently, she is guiding 6 research scholars for their doctoral degree.
Welcome to the UGC lecture series on the topic of polymers in drug delivery, part 3. In the earlier parts, especially in the part 2, we were discussing about the different methods of polymer release, the drug release using the polymer carriers. Especially we entered the topic with the two important types of erosion that can take place when a drug is being carried by a polymer carrier. They are bulk erosion and surface erosion. A little bit note about this two types of erosions will be dealt once again today, and this will be followed by the different advantages and disadvantages of making use of such drug delivery systems. And then we will look into the note of the ideal drug delivery systems. What are the various salient characteristics of a drug delivery system to possess or to have ideally. And then we will be moving on to the goal of the more sophisticated drug delivery systems. What are the primary targets or the achievements, or what is that basically required primarily to have a well sophisticated drug delivery system. This will be followed by a process called micro encapsulation, how the encapsulation of the drug is taking place inside the polymer matrix or the polymer network, and how this polymer can carry the drug. Primarily there are two methods by which the drugs are carried by such polymer materials, and the process is called micro encapsulation. We will be talking about this process, and then we will be moving on to what are called microspheres and the types and the release mechanism that is taking place when such microspheres are being used for such drug delivery applications. Then what are polymer scaffolds and how they do play a role in releasing the drug as well as the mechanism behind using such scaffold materials. And then finally we will be touching upon the various methods that are available or being followed, or the techniques being followed in order to have a controlled and a sustained drug delivery. Now let us move on to the biodegradable polymers, which we have discussed in the part 2. The various examples we have seen how they are being used for delivering the drug by two important modes or methods. One is called the bulk erosion method, the other one is surface erosion. The drug delivery from the bulk eroding method is indicated in the picture here. This is the polymer carrier with the drug getting encapsulated in the polymer carrier, and on hydrolysis or on the polymer degradation when it happens in our body, it releases the drugs like this in the bulk manner. That means it is being carried out from the inside of the carrier, and simply they come out, they get released immediately when a proper condition is getting achieved in the body system. This is called bulk erosion or bulk eroding. The other method can be represented by the B figure, where the polymer is as usual the carrier for the drugs, whereas the drugs they are allowed to get eroded or come out or diffuse out from the polymer carrier only on the surface. So, this is a more predictable and more controllable system where the erosion takes place only on the surface of the polymer carrier, and there is a continuous and sustained release of the drug taking place from the surface. So, these are the two important methods or modes of delivery of the drugs through the bulk eroding and through the surface eroding systems. These are the pictures of different polymers that are being used for such surface eroding. For example, this particular picture shows the surface eroding taking place on the polyarthoester rods that have been put into our body and left for more than 9 weeks. So, after 9 weeks you can see the picture of this polyarthoester polymer, which is basically a biodegradable polymer which undergoes a surface erosion, and the drug molecules would have diffused out like this. After some more time, the right picture shows the condition after 16 weeks of implantation into our body. So, further erosion takes place on the surface and the size of the polymer carrier is going to come down, or it is getting reduced over a period of time, which slowly allows or diffuses, makes the drug to get diffused from the surface. Here are some of the major companies that are involved in the polymeric delivery technology. ALSA, the drug carriers they are called Duros and Oros. Alkermes, they manufacture the ring caps. Nobex Corporation are involved in the development of drug polymer conjugate systems for such delivery technology. Elan, which manufactures Modas and Prodas. Andrex, that manufactures Scott and DPHS. All these are commercial products that are available in market. Now let us have a look into the various advantages of making use of such drug delivery systems, followed by the disadvantages. Initially, the advantages of making use of drug delivery systems, especially a biodegradable polymer as a carrier for the drug. The advantages are less fluctuation in the drug blood levels. That means there is no high fluctuations happening in the level of the drug that is being put into our blood system. It does not go beyond the maximum limit or the toxic limit, or it does not come below the minimum effective limit. So, it stays in a sustained manner, in a controlled manner. It is well within the far extreme limits, and there are less fluctuation when you are going to make use of such delivery systems. Secondly, the frequency reduction in dosing. The number of reductions in the dosing, as we have seen in the earlier episode part 2, where I have shown you the graph. In the case of traditional delivery system, you need to give the dose as soon as the drug is getting over in the body, whereas since you have a sustained release from the polymer carrier, there is no requirement for dosing the drug at a particular period of time. It is there from the beginning to the end with a normal capacity. So, it is in turn reducing the number of frequency or number of attempts to give the drug into the body system. And then the improved patient convenience and compliance is also very important, because of the slow release of the drug. There is no initial burst, or there is no initial amount of larger concentration of the drug which is being forced into our body. So, because of the sustained release, the patient feels very comfortable in such drug delivery systems. And then the increased safety margin of the high potency drugs, especially when we make use of drugs which are highly potent or which has got high potential, they cannot be released in a large quantity. They will have to be sustained and released. So, such cases, these systems play a very important role. Also, the reduction in total health care cost also is another advantage of drug delivery systems. Now, pointing out the different disadvantages of making use of drug delivery systems. The first one is decreased systemic availability in comparison to the immediate release, as it happens in the conventional dosage forms. In the traditional methods, there is an immediate, when there is an immediate requirement for a drug, you can immediately give into that and it is going to get into our body, whereas there is no a situation or condition where you are implicating or where you are putting the drug as a whole concentrated form. So, here there is a decreased availability or decreased systemic availability when compared to the normal conventional traditional method of supplying the drug into the body. The second disadvantage is poor in vivo in vitro correlation. So, this is nothing but when you try to research or work on such polymer carriers as a drug carrier in the external environment, in the outer environment without the body systems, human systems, they will give some result. Based on the result, based on the release profile, drug incorporation into the polymer, we decide what is going to happen within our body, that is in the in vivo system. So, sometimes there may not be a perfect correlation between the experiments that are conducted outside, that is in vitro, and the in vivo processes. So, such things will have to be overcome. Then the possibility of dose dumping, this is another problem which can be associated with such drug delivery systems. And then the retrieval of the drug is also difficult. You cannot simply retrieve it out whenever you feel like. When it is put into the drug carrier, it is there forever and it does its process over a period of time. And then the most importantly, higher cost of formulation. All these biodegradable polymers which are being now used commercially or being tested in the research scale, they are synthetic in nature and they will have to be prepared or formulated, and a separate processing technology will have to be developed, and definitely this is going to involve cost factor. This is the least disadvantage that can happen in the case of drug delivery systems. Now, let us move on to the ideal characteristics or the important requirements for a drug delivery system. How would one want a delivery system to work with, to be in an ideal condition? First of all, basically it should be an inert product. That means the polymer carrier, the material should not in any way react chemically with the drug which is going to be encapsulated or entrapped into the polymer system. So, it should be completely inert or completely chemically inert and should not have any kind of interaction with the drug which is being carried by it. This is a prior or primary requirement of the polymer that can be used in the drug delivery system. Secondly, it should be biocompatible. That means when it is going inside our body, there should not be any kind of interaction once again between the blood system and the polymer. There should not be any other kind of reaction that can create any other secondary effects in our body system. So, it should be compatible to our body, biocompatible. Also, the products which are going to come out from the degradation of the polymer should also not be a toxic material. That is why we have gone for what is called the biodegradable polymers, wherein they are the materials or polymers which on hydrolysis or on degradation come down or break down into smaller and smaller biocompatible molecules which can be excreted out of the body through biological or metabolic processes and happenings in our body. So, in any way they should be compatible to our system and should not create any toxic problem. They are the major primary requirements for a biodegradable polymer to be well used in an ideal manner in such delivery systems. Then comes the mechanical strength. Mechanically, yes, they will have to be strong enough. They should not immediately undergo a loss, or they should not be degradation that is happening in a very short time, because we are always interested in releasing the drug in a sustained manner, in a controlled manner over a period of time. So, it should be potentially stronger to carry the drug as long as we want the drug to be involved or inside the polymer carrier. Then it should also be comfortable, convenient to the patient. This is another requirement. Then capable of achieving high drug loading. The polymer basically should have the basic capacity to load more amount of drug, that is called drug loading. The amount of drug that can be loaded into the polymer carrier, you call it as loading, drug loading, and this capacity of such polymer material should also be definitely higher in order to convey or transport the drug in the required quantity. Let us have a short break and then continue with the other ideal characteristics of the drug delivery. Welcome back after the short break. We were discussing about the different ideal characteristics of a drug delivery system. It is continued. The next ideal requirement is it should be readily processable. Yes, in addition to the formulation, in addition to the drug loading and other basic requirements, it should also be possible for the material to be processable easily by making use of some conventional techniques. The processing of the material into the required shape and size should be feasible, then only it becomes an application part in order to carry the drug and so on. Next important requirement is safe from accidental release. This is what I mean by at once bursting of the polymer, which is going to release out the drug at once immediately, which may be going beyond the toxic limit maximum desired level, and it can create a problem. So, at any cost the polymer should have the capacity to not release the drug or not break down at any point of time to completely release the drug. This is especially very important in such drugs which are going to be toxic when they are going to be taken in higher concentration in our body. And then simple to administer and to remove from the body. That means it should be a simple way to put or administer the drug into our body, and also it should be a simple way of removing the byproducts or the products which are obtained in the lateral stage because of their degradation from our body system. All these things should happen naturally without any difficulties. Then easy to fabricate and sterilize. Yes, in addition to the processing requirement or the fabrication of the polymer drug, it should also be possible to sterilize the polymer samples or the carrier, which otherwise may take any contaminants into our body. That should again be avoided to avoid any side effects or risks. Then comes free of leachable impurities. It should not take along with it any kind of impurities or foreign matters or any other substances along with it, and if this is going to happen, they can leach out and then cause any other problem. So, it should be completely free from the incorporation of such leachable impurities into the system. So, these are all the various ideal characteristics of a drug delivery system. Now, what is the basic goal of the more sophisticated drug delivery systems or the techniques that are available for the day? They should deploy or organize, you can say, to reach the target site in order to limit the side effects. They should not lead to any side effects. Routes the drug through specific areas of the body without any degradation. It should be possible for the polymer to get routed through our body system without getting degraded during its path, and should reach the target site, and at the target site or at the place of requirement the release of the drug should be happening. This should be possible in the case of the polymer carrier. Then comes maintaining a therapeutic drug level for a prolonged period of time. Once again, for a period of time, longer period of time, it should be possible to maintain the level of the drug just at the right proportion, not reaching the maximum or the minimum level. It should be at the right level of releasing the drug over a period of time. Then some other goals are predictable, controllable release rates. The kinetics of the release should be well known or understood by us, how it is happening and what is the rate of release, and then reduce the dosing frequencies and increase the patient's compliance. All these important features are the goals of the more sophisticated drug delivery systems that are being utilized today or in the future also. Now, a little more information about the drug delivery systems. The earliest drug delivery system, first introduced in the year 1970s, was basically based on such polymers that are formed from the lactic acid, glycolic acid and other natural polymers. However, now a number of new polymeric and synthetic polymeric materials are being utilized and also provide the most important avenues for our various research activities. The primary reason for being the polymers being chosen for such applications is that The most importantly, ease of processing, and the second important thing is the ability to readily control the structures, the physical and the chemical properties of the polymers through various molecular synthetic procedures. So, these are the two important reasons. The very basic reason is polymer can be easily processed, fabricated, and secondly you can play differently using the molecular methods to bring or have a control over the various chemical and physical properties that will have to be attained by the polymer system as required for our various applications. These two are the important reasons which has paved the way for the polymers to be utilized in this field very effectively. Now, let us move on to microspheres. What are microspheres? Basically, two broad categories of polymer systems are being used generally, and such systems you call them as microspheres. This is based on their size and the shape have been studied very extensively, and especially based on the dimension, that is size and shape, microspheres can be of two types. One is called the reservoir devices, the other one is called the matrix devices. The difference between these two encapsulation or microspheres are that in the case of reservoir devices, the method involves the encapsulation of a pharmaceutical product within a polymer shell. That means a polymer shell is there and the core is going to be the drug. The drug is going to be the core which is encapsulated within a polymer shell. Then such a device you call it as reservoir device. The second device is called the matrix device. In this case, a drug is physically entrapped within the polymer network. That means you have a matrix or the bed or the base of polymer material into which the drugs are dispersed and throughout it is available in the system. So, this is another way of encapsulation. Both of them are called micro encapsulation, and both the spheres you call them as microspheres. It could be either a reservoir device where the drug is encapsulated with a shell having the drug as a core, or the drug can be dispersed and distributed in the polymer matrix where you call the device as matrix device. So, this is a picture which shows you the encapsulation of the microsphere. So, this is your polymer carrier which is about 100 micrometer, that is why it is called as microspheres, and the drugs are inside that, it is encapsulated within the polymer system. Now, let us have the definition for such a micro encapsulation. It is a process that involves the encapsulation of protein or peptide drugs in small porous particles for protection from any other external agencies, that is insults, and for sustained release. So, it is a process of encapsulating the drug into the polymer system which is protecting it from any other external agencies, at the same time slowly allows the release of the drug from the encapsulation system. Now, there are two types of microspheres that are being basically used in such applications. One is called the non-biodegradable system, the other one is biodegradable systems. Non-biodegradable systems are the methods where you make use of non-biodegradable materials like ceramic particles, polyethylene co-vinyl acetate, polymethacrylic acid, polyethylene glycol combinations. All these are examples of non-biodegradable microspheres, but the most preferred and the important ones are biodegradable microspheres, which are made of such polymers like gelatin, polylactic co-glycolic acid, shortly called as PLGA. This is a copolymer of lactic acid and glycolic acid. These are the examples of such preferred biodegradable polymers which are used as microspheres. So, this particular picture tells you how the entrapment or the encapsulation is taking place by two different modes. During the year 1970, scientists began to encapsulate and entrap the drugs within the polymer systems. Encapsulation involves surrounding the drug molecules with a solid polymer shell which is nothing but the carrier. So, this is what is shown in this picture. The drug is inside the polymer shell. So, such a device you call it as a reservoir device. The drug is carried by the polymer shell. The other type is entrapment is involved with the suspension of drug molecules within the polymer matrix. This is the second method where you call it as the matrix device. So, this light blue indicates the polymer carrier into which the drug is dispersed or suspended throughout the polymer matrix. Now, this device you call it as matrix device, which is different from the reservoir device. Whatever the method could be, it could be a matrix device or a reservoir device. Both of them are called encapsulation systems, and both of them undergo what is called the drug release by the diffusion process. So, what is diffusion? How it is happening for the release of the drug? We are going to see now. Early encapsulation entrapment systems released the drug from within the polymer via the route of molecular diffusion. What happens in such molecular diffusion is that when the polymer absorbs the water, it basically swells in size initially, and the swelling creates large number of voids throughout the interior of the polymer, and the smaller molecular drugs which are existing inside the entrapment, that is inside the polymer, can escape via these voids that are developed at a known rate that is controlled by the molecular diffusion. So, this kind of diffusion you call it as molecular diffusion, which releases the drug into the body system. So, this is the schematic representation of such a drug release happening by molecular diffusion. You have the polymer carrier, drugs are inside, add water, hydrolysis or polymer degradation happens, voids are created, it swells initially and the voids are created. Because of the voids created, the drug is able to get diffused outside. This is the mechanism shown in the schematic way by diffusion method. Now, the microsphere release. The microsphere release can be hydrophilic, hydrophobic or a combination of both called hybrid. If it is going to be the case of hydrophilic, that is attracting the water, for example, gelatin is used for such a purpose, it is best suited for the burst release, that is immediate release, which may be due to the bulk erosion of the material, that is your gelatin. The second release could be due to hydrophobic nature. This is explained with the best example of polylactic glycolic acid. It is a copolymer which is very useful in the good sustained release of especially the vaccines in our body, because it undergoes of course a bulk erosion, but at the same time it has got a sustained release of the drug. But the disadvantage of using such a polymer in these systems is that they tend to denature the proteins, especially when proteins or peptide drugs or amino acids, they are used as the drug, they get denatured or they get degraded, and this effect is one of the most important disadvantage of making use of such hydrophobic PLGA polymers. The other could be the combination of both, that is hybrid system, otherwise called as amphipathic systems. Here we possess both, that is good sustained release is also possible. At the same time, it keeps the proteins and the native active inside the body, inside the carrier without degradation. Then comes the polymer scaffolds. Polymer scaffolds are the polymeric matrix into which the drugs can be easily incorporated. Protection of the drug is very much possible when you are going to make use of the polymer scaffolds from any enzymatic degradation. It is applicable to especially the peptides and protein drugs. The released drug at controlled rate over a longer period of time is also possible using such matrix, that is polymer scaffolds. The drug is dispersed or dissolved in a suitable polymer when they are used as the entrapment form. Release happens due to the diffusion of the drug through the polymer and diffusion through pores or voids that are developed in the polymer structure. Therefore, different release profiles can happen by making use of such polymer scaffolds. They could be a result of either the dissolved or the dispersed systems. So, as a summary, what we have seen in today's part includes the drug release by bulk and surface erosion, advantages of drug delivery and disadvantages of drug delivery systems, various ideal characteristics of drug delivery systems, and then the goal of more sophisticated drug delivery systems. This was followed by the process called microencapsulation, microspheres and their types we have seen, and then finally the polymer scaffolds. Now I have few questions. What are the merits and demerits of drug delivery systems? List down the characteristics of an ideal drug delivery system. What is the ultimate goal of a sophisticated drug delivery system? Name few polymers that may degrade within our body. What is microencapsulation? What are polymer scaffolds and what are their requirements? What are the types of microspheres? Thank you.